Assalamualaikum, my name is Nur Aina Maisara U2100625 from HJ2 will present about the evolution of eukaryotes. Living things have evolved into three large clusters of closely related organisms called domain archaea, bacteria, and eukaryotes. As life on Earth started to undergo evolution and become more complex, the simple type of cell called a prokaryotes underwent several changes over a long period of time to become eukaryotic cells. Eukaryotes are more complex and have many more parts than prokaryotes. It took several mutations and surviving natural selections for eukaryotes to evolve and become prevalent. Scientists believe that journey from prokaryotes to eukaryotes was a result of small changes in structure and function over very long periods of time, somewhere between 1.5 to 2 billion years ago. The most widely used identification of a eukaryotic cell is the presence of a nucleus, which is to store the genetic information and DNA of the cell. In the prokaryotes, the DNA is just found in the cytoplasm, usually in a single ring shape, but eukaryotes have DNA inside of a nuclear envelope that is organized into several chromosomes. Once the cell had evolved a flexible outer boundary that could bend and fold, it is believed that the DNA ring of the prokaryotes was found near that boundary. As it bent and folded, it surrounded the DNA and pinched off to become a nuclear envelope surrounding the nucleus where the DNA was now protected. It was a favorable adaptation, so DNA is not tangled or unevenly split during mitosis or meiosis. Now that the nucleus had appeared, other internal membrane systems like the endoplasmic reticulum and the Golgi apparatus evolved. Ribosomes, which had only been of the free-floating variety in the prokaryotes, now anchor themselves to parts of the endoplasmic reticulum to aid in the assembly and movement of proteins. Research conducted by Lynn Margulis developed endosymbiotic theory, which is widely accepted. This theory stated that eukaryotics may have been a product of one cell engulfing another, one living within another, and evolving over time until the separate cells were no longer recognizable as such. The endosymbiotic origin of mitochondria and chloroplasts is widely believed because of many similarities between prokaryotes and these organelles. Both mitochondria and chloroplasts are similar in size and shape to prokaryotes, have their own circular DNA attached to the inner membrane same as the prokaryotes' DNA, but the DNA sequence do not match the DNA sequence in nucleus. Their ribosomes are more similar in size to prokaryotic ribosomes. Two or more membranes surround these organelles and they divide by binary fission instead of mitosis. Therefore, they appear to be more like prokaryotes than eukaryotes. Mitochondria derive from engulfed aerobic prokaryotes, specifically an alpha proteobacterium. Rather than digesting them, the bacteria remain as symbionts, benefiting the host cell by giving the ability to use oxygen to release energy stored in nutrients. Most mitochondria are shaped like alpha proteobacteria and are surrounded by two membranes, which would result when one membrane-bound organism was engulfed by another membrane-bound organism. The mitochondrial inner membrane is extensive and involved in foldings called cristae, resemble the texture outer surface of alpha proteobacteria. The matrix and inner membrane are rich with the enzyme necessary for aerobic respiration. Eukaryotic cells may contain 1 to several thousand mitochondria, depending on the cell's level of energy consumption, with each measure 1 to 10 micrometer in length. This enables eukaryotes to make more efficient use of food sources than their prokaryotic counterparts. Some groups of eukaryotes, such as plant cells and some algae, are photosynthetic, which undergo photosynthesis. Their cells contain another kind of organelle called chloroplast that rich with pigment chlorophyll involved in converting light energy into energy-storing sugars such as glucose. Scientists believe that chloroplasts evolved from photosynthetic prokaryotes, cyanobacteria that live inside the cells of an aerobic heterotrophic eukaryote. Chloroplasts of primary origin have thylakoids, a circular DNA chromosomes and ribosomes like those cyanobacteria, and each chloroplast is surrounded by two membranes. In conclusion, Evolution of eukaryotes about 2 billion years ago explained by endosymbiotic theory. Mitochondria and chloroplasts evolved from prokaryotic organisms are not free-living bacteria anymore, as these organelles have become completely dependent on their host cells since the evolution of first eukaryotic cells. The difference between eukaryotic cells and prokaryotic cells is eukaryotic cells have many types of membrane-bound organelles that divide the cells into many different compartments, such as nucleus, which holds genetic information in the form of DNA, endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria to create ATP molecules from glucose, and chloroplasts to create glucose from sunlight in plants. Prokaryotic cells have one type of organelle, which is ribosomes, but these organelles are not covered in membrane, and the DNA of prokaryotic cells simply float around the cytoplasm.